Yeah, so we test weekly at 5 beaches in Isla Vista, as well as Hendry's Beach right here. And we will incubate on Friday, then read the samples and share the results on Saturday. What we do is we test the water for the level of Enterococcus bacteria, which indicate fecal pollution. The greater the Enterococcus level, the higher the risk of human health problems. Specifically for the process, we dilute the water sample, add a reagent, which is basically food, seal it in a Quantatray, and then incubate at 41 degrees Celsius for 24 hours. To read, we use a UV light to spot any fluorescent wells, which help us calculate the most probable number of Enterococcus per 100 milliliters, because it's just a number we use to estimate. So the California State Water Resources Control Board has set water quality standards that we compare our results to. For example, if the most probable number per 100 milliliters is greater than 104, we would highly advise the public to limit their water activities like swimming or surfing. Enterococcus aren't harmful on their own, but they can indicate other, more dangerous microorganisms and viruses. Usually this fecal bacteria can come from human or animal waste or sewage or stormwater runoff. So if we had a really high number, we might estimate that the beach was down from like a runoff drain or a dog park. Yeah, there's a lot that both people and groups can do. On an individual level, we recommend encouraging beachgoers to clean up after their pets and take their trash with them, as well as doing activities at their home to reduce water runoff. This includes not wasting water indoors, fixing oil leaks in your car, and improving landscape techniques. Surfrider has several resources online with more specific information that I can share with you guys. On a more community level, it's also important to look at the organizations in your county and city. I think it's important to know what the main water and sanitation companies are and where they're located, as well as any farms and factories that could potentially affect water quality. Yeah, so overall, California has fairly healthy beaches, so Santa Barbara County as a whole is already doing great. According to a report from Heal the Bay that surveyed over 500 state beaches, they estimated 92% of beaches had good water quality in 2019, with the state's 10 dirtiest beaches residing mostly in San Mateo County in Northern California, and then 4 different specific spots in Southern California. So yay, Santa Barbara.
Hi, my name's Claire. I'm with Surfrider's Blue Water Task Force, and I'm a third-year biology major at UCSB. And today I'm here to read the samples that my team member Mia incubated yesterday. So basically what we're looking for is Mia fed the bacteria that we're looking for. She put those little pellets in the water and shook it up, and they've been nice and warm inside of this 35 degrees Celsius incubator overnight. And so if there were any bacteria there, they've been given a chance to grow, and the way that we'll tell if they're there is I'm going to check them with this black light, and if they glow under the black light, then we know that they're there. And depending on the number of little squares that glow, we can make this calculation using this table. It's kind of like a multiplication table, and that allows us to make some really educated guesses about the bacteria count in the water. So let's check and see what we've got. The first thing that I'm going to check is our control. So if our control is clean, then that means that none of our samples were contaminated, and whatever we find is for real. It's a reflection of what's going on out in the water. Okay, so it looks like our control is clean, so I'm going to go ahead and check the rest of our samples, and I'll know that they don't have any accidental human-introduced bacteria in them. Okay, so that looks good. I just checked Devereaux Beach, and then right next to Devereaux in Isla Vista, we also test sands because they're on 2 sides of a point. And so we like to see if sometimes there's differences in the water quality on either side of Coal Oil Point. Okay. Alright, that one's clear too. They're all slightly toasty. It's like I just took them out of the oven. Looks like it's also good. So I'm just trying to create like some darkness with my body so that when I scan it, if it were contaminated, it would glow really brightly and it'd be super obvious. Small wells and big wells, and those are calculated differently if we were to see any of them glowing. They tell us different things about the numbers. Cool. Okay, so on our sample trays, which are sealed in our Quanti-Tray sealer, and then we label which beach they're on, we have these small wells and these large wells, and depending on which is fluorescing, it changes the calculation that we make about how many colonies are likely to be in the water. And then this long cell up here is sort of our overflow. So as this packet gets sealed, if there's extra water in the sample, it just gets pushed up here. And then finally we have Hendry's, which is where we are, the Water Resource Center. So we just started doing this one since we've moved our lab equipment down to the Water Resource Center, who were very kind to let us share their space, and we wanted to see what was up locally. So we're checking here in the dark just to double make sure, and it looks like we actually have, on closer kind of higher contrast inspection, we have one cell there that's probably got a little bit of bacteria in it, and this is our, so we went back and looked in the dark, and we found that one of these small wells was slightly fluorescing on the Hendry's sample. And so with that information, we'll be able to put it into our chart and get a number. Yeah. So based on our findings of one small well fluorescing at Hendry's, we can then reference our MPN table and check for the point where 0 large wells and 1 small well cross on the chart, and this allows us to derive our MPN number, which stands for the most probable number. So this is a metric of statistically how many bacterial colonies are likely to be present in the water. And in this case, because only 1 small well fluoresced, that number is pretty low. But when it gets higher, it's a really helpful metric for us to be able to share with the public to kind of gauge the risk level of going out on any given day.
Hi, my name is Mia Lombardo, and I'm a 3rd year at UC Santa Barbara. A citizen science program is any program or project that encourages public participation and collaboration in conducting scientific research. What that means for us as part of the Blue Water Task Force is that we collect water samples at 5 beaches in Isla Vista, and then we test them, and then we get to share the results with Isla Vista residents. Citizen science programs like the Blue Water Task Force are special in that they usually cover areas like beaches that aren't covered by state or local agencies and focus on more specific problems. Yeah, there are a lot of great opportunities and organizations if you're interested, like Surfrider, if you're interested in just like beach conservation and preservation. The chapter that I'm a part of in Isla Vista and Blue Water Task Force itself is student run and it is fairly insular, but we also have a lot of beach cleanups and other events and just a lot of other tips and stuff that we post on our website, Facebook, and Instagram. Yeah, and then the Santa Barbara Surfrider chapter itself also has a lot of great events. I don't think they're doing meetings or any in-person events right now, but be on the lookout. And also Santa Barbara Channel Keeper is another great conservation organization that does the same, like water quality monitoring and other things. So if you're interested, there's so many cool things to do in Santa Barbara. I just really want to emphasize that Surfrider is a really cool organization. I hadn't heard of it before going to college, and this is now going on my 2nd year as part of the Blue Water Task Force. I just really love getting to actively be part of the community through testing and monitoring water quality, and then getting to share those results so that other people feel more safe and confident about the waters they swim and breathe and play in. Mm-hmm. I think it's really cool because a lot of these chapters, especially ones that are run like fully by students like we are, it's like we're not doing it for profit, we're just doing it because we all have a vested interest in our oceans.